As usual, we have our CPMS slides at the very beginning, so you can read through the information on the slides. We have launched our new clinical patient management system. So if you're interested in uploading a clinical case for advice, then contact us via email. We will be more than happy to help you.
Good evening and welcome to the eUROGEN webinar series. I'm Michelle Battye, the eUROGEN Programme Manager. And just before we get started, I'd like to take a couple of minutes to explain just a little bit about what ERNs are and do. So European reference networks were created and they're funded by the European Commission since 2017. There are 24 ERNs covering most medical fields, and ERN EUROGEN, we deal with rare urorectogenital diseases and complex conditions. So they're all areas that need highly specialised surgery where the expertise is rare. And we are currently a network of 56 healthcare providers in 20 member states. So a little bit about what we do. Well, as you saw, I hope, from the slides at the beginning, we do virtual cross-border consultations using the clinical patient management system, which is a secure tool provided to all the ERNs by the European Commission. So please feel free. We've just launched CPMS 2.0. So really, I would strongly encourage you to get in touch if you do have a tricky case that you would like to discuss with our network of experts. We also do education and training activities like this webinar series, and you can find them all on YouTube. So please go and have a look at the past ones. And we also collaborate together with scientific societies on producing clinical guidelines and clinical decision support tools where there's less evidence. And then finally, we are developing a registry, which is very exciting. We expect it to be very important for future research. We all know that data on patients with rare diseases is scattered and fragmented. So we very much hope that our registry, when it's fully rolled out across all our healthcare providers, will be a really important tool, both for research and for the evidence for the clinical guideline development. And we currently have about half of our hospitals in the registry entering patient data, and we have just reached a milestone of having over 1,000 patients. So again, a strong encouragement to all our healthcare providers. Please start to enter patient data, and also to all the patients out there, it's incredibly important that you please give your informed consent. By doing so and being in the registry, you're findable if you want to participate in future research, such as clinical trials. And obviously, the bigger the data set is, the better research we can conduct. So tonight's webinar is part of our colorectal series, and it's joint with ERN Ernica. And we're very happy that it's also supported by EUPSA and ESPU. And I also want to mention that you are eligible for a EACCME accreditation for live attendance at this webinar if you fill in the survey within 2 weeks. So our speakers here this evening, they're going to talk about the dorsal cloaca and its fate, key in the formation of ano-rectal malformations. And we have two speakers this evening. We have Jill Hikspoors, who is Assistant Professor at the Department of Anatomy and Embryology, and she's at Maastricht University in the Netherlands. And her research focuses on the establishment of an interactive, three-dimensional topographic atlas of prenatal development. So we're really looking forward to hearing more about that. And then we also have Ruben Visschers, who is a pediatric surgeon and gastro-intestinal surgeon, and he's in Maastricht University Medical Center and Uniklinik in Aachen. And his areas of interest ranges from congenital disorders such as ano-rectal malformations and Hirschsprung's disease to acquired conditions such as inflammatory bladder disease. So really huge thanks to both our speakers this evening. Thank you very much for joining us and sharing your expertise. And Jill, I give you the floor. Thank you.
Thank you very much for the invitation and also for the introduction. So let's start. Well, I will start with introducing you to what is the cloaca and what is its fate. And then I will show you how the pelvic floor muscles are developed, as does at the area where the cloaca is as well. And at the end, I will start brainstorming with you to see if we can up with the time window. So when do these ano-rectal malformations develop? And can we predict the difference in severity from an embryonic perspective? So first of all, what is the cloaca? Cloaca is the common exit of the urogenital sinus and the anorectum, which we can show it schematically, histologically, and also in 3D. And it's mainly the 3D shape, which we built from these high-resolution histological sections that allow us a detailed observation of the cloaca, which you see here with the yellow asterisks. And mainly what we can do with the models is that we also can use these 3D. So what is the general concept of this cloacal subdivision? If we go back in the very past, in 1888, Tourneau was the one who first explained this concept by an active frontally displayed septum that would extend in a caudal direction, which you can see here with the red arrows. So from one common exit, the idea is that you will end up with two tube-like structures. What he did not do was that in lateral direction, how his extension of the septum looks like. And that's what Rathkes added to this active concept of eUROGEN septum development. If we look to the histology on the right, we do see that Rathkes' plica are suggested that from lateral to medial, there is a fold that allows the tube, which in picture 1 is still a sort of A shape, but at least one lumen, whereas in picture 2, you can see that we have two tube structures, which shows you that the cloaca is subdivided completely. And all this happens between 5 and 7 weeks. But what will be the underlying mechanism? As recently, or 30 years ago, Van der Putte claims that it's not an active process as the eUROGEN septum is extending in caudal direction, but it's a passive way due to the straightening of the body axis. And while this concept is it's not very much debatable, it's also not quantified yet. So only due to his observation of straightening of the body axis, it's now believed to be a passive concept of eUROGEN septum development. And with our study, because of the 3D models we have, we can also quantify this part. So in order to do so, we collected human specimens, we digitized these histological sections, and by stacking these sections on top of each other with our Mira 3D software, we could align these embryos back into its 3D shape and reconstruct the structures of interest. And this allows us then having these 3D observations over time so that we can visualize the difference in shape of the cloaca and the subdivision but we can also start to quantify this. So let me introduce you to the schematic models that we got from this. In order to have an idea about the cloacal development, we started with recognizing landmarks. And what we found was during early embryology that the Wolffian ducts, which is shown in blue here in these pictures, start from 31 days to insert the cloacal region at the sacral level number 3. And over time, it does not change anymore. So it remains a stable landmark. What we also have seen between 4 and 5 weeks is that the body is folding. So there's a caudal elongation of the body axis. And what we see is that in orange, the allantois, which is different in epithelial thickness. So that's how we could distinguish that component. And on the dorsal side, we do see in green the gut tube, and where the Wolffian ducts and the peritoneal pouch, given with the asterisk, remain at the same position, we do see together with the folding of the body axis that the brown component, which is the common exit, so the cloacal region, that one is extending as well in caudal direction. So that's between 4 and 5 weeks. And what happens after that? After that, septation starts, and what we do see is that from the eUROGEN septum in a ventral direction, so from the asterisk downwards towards the red, which is the urethral plate, there we do see an exponential extension in caudal direction, whereas on the back side, where we do see the light pink in the 3D models, we have the dorsal cloacal membrane, which is a very thin layer of skin only. There is less growth visualized. So that means indeed that the mechanism of Van der Putten, who says that on the dorsal side there's a different growth rate than on the ventral side, is causing a subdivision of the cloaca into 2 tubes. This all happens between 5.5 to 7 weeks. In order to see where this differential growth is specifically, we did a lot of measurements, from which I will show you a few examples. What we first checked again, of course, was the position of the Wolffian duct over time. So in the red dotted line, we can see indeed over time between 30 and 70 days, so up to 10 weeks, we never have seen a change in the sacral level of the Wolffian duct. Whereas in a very short time period, the solid red line makes a steep decrease in caudal direction, so meaning that the eUROGEN septum indeed is extending in a dorsal area. Then we used the Wolffian duct to see what happened on the ventral side with the cloacal region, so the brown structure. With the dotted blue line, we can see that the proximal area proximal to the Wolffian duct is not growing. If you check the graph, it over time remains the same in distance, whereas the part from the Wolffian ducts up to the urethral plate, we do see that is the same rate in growth as the body axis. The body axis is shown over here with a solid black line. And what we also checked is from the distal tip of the eUROGEN septum towards the ventral tip of the urethral plate, we do see indeed that also here we do see an extension with the same rate as the body axis, whereas the dotted purple line towards the tip, so towards the dorsal cloacal membrane, that part is not growing. And with these signs, we could see specifically where this differential growth is then located and causing its subdivision of the cloaca. So what we do see with the 3D models on the right side, we do see a growing zone on the ventral side and of the gut tube itself, but at the space in between where the septum becomes positioned, there we do see a growth, the zone which is not growing. And that's the dorsal tip of the gut tube. So in summary of this first item, the cloacal subdivision is determined by a ventral growth zone and a dorsal not growing zone. And this process is during the straightening of the body axis happening, so between 5.5 and 7 weeks. If we then check what happens after that, so what is the fate of the dorsal cloaca, we also did see a lot of changes. At 7 weeks, the brown area, and also histologically shown here with the pictures 1 and 2, we do see that it's a very narrow lumen, which is almost like only 1 cell in diameter large. And that's the tip. At 8 weeks, we do see it has a more balloon-like structure, so it's swollen. And at 10 weeks, we do not identify the cloacal region anymore. How can we then distinguish what is the cloacal region? We identified also landmarks for this component. What we do see if we think about Hilton's white line, also better known as the intersphincteric groove, which is the part proximally we have the levator ani muscle, and caudally to it we have the external anal sphincter. So what we do see is that proximal we have the gut tube covered with a smooth muscle coat, shown here with the green arrowheads. And you do also see in 7 weeks in the 2D picture that the part in green, that's covered by this smooth muscle coat, which is the brown area. That's the area in the histology as well visible, which is not covered by a smooth muscle layer. And at the bottom part where we have the peach color arrowhead, there we do see the multi-layered squamous epithelium. So that's the pectinate line. What we see over here, that's the endodermal to ectodermal transition. And if we look with these landmarks to the 10-week situation, we do see that the intersphincteric groove between the 2 muscles and the pectinate line, so the transition between endo and ectoderm, is at the same level. So we do not have an area of endoderm anymore without a covering of the smooth muscle. So we have to think about, is the cloacal region then embedded in one of those regions, or what happened with that area? And when we look over here, we also do see that the smooth muscle coat ends up to the external anal sphincter, forming at the bottom part also the internal anal sphincter. So what happened if we check proximal to this intersphincteric groove? We do see that during the disappearance of the cloaca, also the epithelium starts to mature with a goblet-rich area and a goblet-poor area. All of that is proximal to Hilton's white line, so the intersphincteric groove, with the goblet-poor region better known as the columns of Morgagni in the adult situation. So that means that we have no cloaca anymore below this intersphincteric groove. We only do see those squamous epithelial cells of the ectodermal origin. So what happens at the moment between 9 and 10 weeks during the disappearance of the cloaca? We do see metaplasia. We do see a replacement of the epithelium. So what we can see at 9 weeks is that we, below the boundary, we still have that balloon-shaped area, but where we also do see that the cells are covering each other. So we have the endothelial cells and the ectodermal cells lying on top of each other, and that region of replacement becomes shorter and shorter until we have the situation at 10 weeks where no cloaca is present anymore. So the dorsal cloaca will not be a part of the adult anorectum. What problem do we have then if we compare the adult situation with this 10-week situation? It's the pectin, because at 10 weeks we do see that the intersphincteric groove together with the pectinate line are at one level, so that we do not have a region in between at this moment. And for that, to get an answer to that, I will first take you with me to pelvic floor development. And if we check pelvic floor development, we have seen that we have 3 types of mesenchyme, or 3 regions, better to say, of mesenchyme. One part of mesenchyme is surrounding the gut area, which we can see in the histology at the HG for hindgut. We do see also that it's quite isolated because it's still embedded intraperitoneally. So that mesenchyme is isolated from the mesenchyme around. We have pelvic mesenchyme, which is isolated by these black line, the dotted line, and we have the mesenchymes that is also more dense, which is for limb development. And the pelvic mesenchyme and the limb mesenchyme can be separated from each other by these area where we have less dense mesenchyme, and that area allows vessels and nerves to pass. And if we go from 6.5 weeks back, then we did see already at 6 weeks that area with less dense mesenchyme, and at 6.5 weeks we also could see indeed that the pudendal nerve, for example, is passing over here, which on its medial side we have the pelvis, and more on the lateral side of the pudendal nerve we have then the mesenchyme, which is for limb muscle development. And what tells us this further, if we check the development of the pelvic floor muscles at 6.5 weeks, we can see that from this light blue mesenchymal mass on the ventral side, so ventral to orange, we have light blue but also purple structure, so that the urethral sphincter and the urogenital swellings are here becoming identifiable. And if we take this off and only have a view on the back, so the ventral area where the development of the bladder is taking off, we do see now from the ventral side the ano-rectum with green and brown, still with the cloaca included in this at this age. And we do see in light blue an area which is quite flat but more vertically oriented, and we do see at the bottom part an area with light blue at 6.5 weeks, which is more horizontally orientated. And at 7 weeks, we do see also that area is more separated from each other. That's the area of the intersphincteric groove, with proximal in orange and in pink, the development of the levator ani and the coccygeal muscles, whereas at the bottom part, the light blue part starts developing between 7 and 8 weeks into the ischio-cavernose, the bulbous spongiose, the transverse perineal muscle, and of course, quite in the back, at 8 weeks, the external anal sphincter in dark blue, but meaning that all those pelvic muscles develop from the same mesenchyme at early development. What can we tell then? If we have 10 weeks still Hilton's line and pectinate line at the same level, how do we end up with this pecten? If we follow the shape of the external anal sphincter, this part has at the beginning a more roundish structure or more vertically oriented structure, whereas if you follow this over time, up to 20 weeks of development, we will see that the angle is really changing. So that means if we have Hilton's line over here, I don't know if you can see my arrow, but together with shaping of the external anal sphincter, Hilton's line will become more near to the exit, whereas the pectinate line, which is the transition between endoderm and ectoderm, will remain at this position where it is at this moment. And that's how we end up with the intersphincteric groove more near to the exit, and having a pecten in between the pectinate line and Hilton's white line. But that takes a little bit of time. The development of the muscles is there at 8 weeks, but shaping and maturation of this has then a longer period needed. So also for this short conclusion, the dorsal cloaca disappears by metaplasia in the 9th week, pelvic floor is complete at 8 weeks, and Hilton's white line becomes more positioned distally to the pectinate line due to the shaping and the maturation of the external anal sphincter, and this causes then the appearance of the pecten. I will also take a short side step to the female situation, as if we want to brainstorm and think about ano-rectal malformations, we also need a little bit more information about the ventral area of the cloaca, not only about the ano-rectum. So what we do see here after cloacal subdivision, so once the body axis is straightened at 7 weeks, the first appearance of the Müllerian ducts become visible, shown here in light blue. And at 8 weeks, we do see that both Müllerian ducts have merged on the midline, and that they insert the ventral area, the dark gray area. Then over time, we do see at 14.5 weeks, we do see that this insertion point is more near to the exit, where you can see with the peach color, the area where we have the exit, so the skin boundary. And if we follow this over time, then we can see at 20 weeks, also again here, maturation takes a long time. So not only focusing on the embryonic period, but also early fetal period, we do see that we have a dorsal extension of the urogenital sinus in order to have squamous epithelium in an upright direction with the cervix at the level of the bladder neck, which is still of Müllerian origin. So we do also here see a replacement of cells at the area of the Müllerian duct, which at 20 weeks has evolved in the vaginal area. And this is how it looks if you would view it from the bottom side. We have the ano-rectum quite far to the back, whereas the axis of the urethra and the vagina are quite close to each other, as in normal development is the case. And if we would like to see how this growth and differences in uterus development here are shown, we have used landmarks, the ureters. We can see that ureters in yellow have an insertion point into the bladder at the level of sacral 2, and they do not move over time. And that was again a stable landmark for us to see how this merging point or insertion point of the Müllerian duct with the vagina and the urethra is evolving. What we have seen is the area between this insertion point and the exit of the, so where we have the skin is not changing over time. You can see that in the graph as the black line, whereas with the red area between its insertion point and the ureters, we do see have a growth phase, so the red line in the graph. And what you can see is a quite steep growing phase up to 10 weeks. And after these 10 weeks, so 70 days, we start to see a more plateau formation of it. So growth is much lower in its speed, but that's the time where maturation starts to appear. So that's the time point where we do see that the Müllerian duct becomes replaced by squamous epithelium in order to form the vagina. And if we would compare this to the male situation, so where we have the development of the Müllerian ducts, and if we would focus on the urogenital area, and also see with respect to those differential growth components, we do see that in order to form the penile urethra, we do see a quite steep ventral growth with respect on the dorsal side, where we had it in the female situation. In the male situation, it grows in the ventral directions. So in conclusion over here, before I go to the next step, we have also in this time window, so we first have a time window with cloacal subdivision where we have differential growth. We have on the dorsal side a non-growing phase. On the ventral side we do see here when we have to make a distinction between the female and the male situation. We have in the male situation again a ventral growth zone, whereas in the female we have a dorsal growing zone in order to generate a vagina. So if we think about ano-rectal malformation, what can tell us all this embryonic information then? What you can see with the schematic picture on the left is that many variations are possible, but if one of these variations must occur, it's, to my opinion, to my idea, based on the argument that I've shown you so far about embryonic development and the fate of the cloaca, it's everything starts at 5.5 weeks with this differential growth. If the zone displayed here with the 0 between the green and the brown structure, if this part is not stopping growing, Then if it does not stop at all growing, then we end up with a cloaca in the neonate. If it grows too fast, but still less than the brown area of growing, then we will end up with an anal exit, which is much more to the ventral side instead of at the anal area where it should be. And if it stops immediately with growing, we will have a blind pouch, so the rectum would not have an exit at all, because then it cannot break through the dorsal cloacal membrane. But to me, the time window of developing an ano-rectal malformation, that's at 5.5 weeks when this would be disturbed. So if this non-growing zone is not properly coordinated, then all these variants can occur depending on how disturbed this region is. And with that, I would like to make a final conclusion, which you should also take home with you, is that the normal development should, the cloaca not taking a part in, with the external anal sphincters part of the pelvic floor mesenchyme, whereas the internal anal sphincters from the smooth muscle coat around the gut itself. Female and male growth, they differ in rate, in formation of the urethra and the vagina as well in the female situation. And to me, all these embryonic information suggest that the anorectal malformation originate at 5.5 weeks due to a disturbed differential growth on the dorsal side of the cloaca. And with that, I would like to thank you.
Thank you very much. We're now moving over to our second presentation from Ruben Visschers.
So, Ms. Hikspoors spoke about the period of life during the embryological development, and I would like to continue this story, knowing that a lot is still to be unraveled. After birth, the neonate with the anorectal malformation, and dive a bit more deep into the musculature and sensory nervous system and the problems that we can see there, or the morphological problems. When we look at the anatomy, it's maybe good to recap. We're dealing with the muscle layer of the rectum, with the internal anal sphincter, external anal sphincter, the puborectal muscle, and the levator ani as a whole. And they don't work by itself. When feces enters the rectum, the sensory nerves send a signal to the spinal cord. A motor neuronic reflex is relayed back to the rectum. It says, please relax everything. Luckily, at the same time, from the cerebral cortex, the voluntary muscles get a signal to tighten the external anal sphincter, and at that moment in time, we have time to find a toilet. Of course, a lot can go wrong, and I won't dive into all the things that can go wrong, but of course we can imagine that in all manners and ways, when it goes wrong, we're dealing with either constipation or fecal incontinence. Well, now, what is shown when we look at the levator ani muscle studied in MRI, that especially in the high imperforate anus type, that the levator ani ultimately is thinner, up to 2 millimeters, that the ratio is more skewed, meaning that there's more asymmetry left-right, and also the angle is significantly different. Do we have some information about the anal sphincter? Yes, we do. With 3D CT scanners They've seen all kinds of morphology, from aplastic, J-shaped, and double ventricle in the high and in the intermediate anal rectal malformation. And for me, most striking actually is that ultimately in this, albeit somewhat limited group, but still 26% of these patients has a normal anal sphincter. Do we know something about the volume? The answer is yes. The volume of the anal sphincter, also measured through MRI, is also lower, corrected for age, and also lower compared to the normal population. And also significantly lower in the patients with an unfavorable prognosis. They also looked at the striated muscle complex as a whole. So that meaning the levator ani, the puborectalis, and the external anal sphincter. And there they looked at the development of that muscle and also divided it into perianal and recto-urethral fistulas. And there you see that up to 3/4 of the patients with a perianal fistula are said to have a properly developed striated muscle complex, whereas in the recto-urethral fistula group, up to 60% is said to have a poorly developed striated muscle complex system. They also tried to correlate that the development of the striated muscle complex with the bowel function scores, and that was a significant correlation, showing that the lower the or the poorer the development of the muscle complex, the lower the bowel function scores, so worse clinical scores. And on the other hand, a proper or good developed striated muscle complex showed higher bowel function scores. They subdivided that into the perianal fistulas and the recto-urethral fistulas, which showed a significant relation again in the perianal fistulas, but it did not reach statistical significance in the recto-urethral group. We could discuss whether this is an issue of patient group size. But ultimately, there is a significant correlation between the striated muscle complex evaluation and the bowel function score as seen here. Previous studies were all performed in with imaging, MRI, CT scanning, but there's also a large study in babies who unfortunately deceased with an ano-rectal malformation, but the cause of death was unrelated to that congenital anomaly. And here you see some normal values, which for me most striking is to see how thin at the left side the internal anal sphincter is, but also at the right side the thickness of the levator ani muscle is. So it's almost 1 millimeter. And the group proposed a system of muscle tubes, and they called one was the intra-anal muscle tube at the left in green. They have the longitudinal muscle tube, which is here the levator ani and the rectal wall muscle, and the transverse muscle tube, which is the puborectal and the external sphincter tube. And you already see, and we're getting that in the next slide, that there are structural differences among the type of anorectal malformation patients. And what do we see? That in the internal anal sphincter tube, which consists of the internal anal sphincter, that it is said to be absent in the anal canal. The longitudinal muscle tube, consisting of the levator ani and the rectal wall smooth muscles, is displaced a little bit upward. You see a little picture at your right, and anteriorly. so it's elongated. The tube through which the anal canal should have passed is closed, there are fewer muscle fibers, and there are no sensory nerve fibers. The transverse muscle tube, consisting of the puborectal and external anal sphincter, also a little bit displaced upward and narrow. That's the red dot at your upper right. But that, in that way, has become shorter. So it becomes also more difficult for the surgeon to position the neo-anus correctly. Fewer muscle fibers and also fewer spindle cells there in the tissue. Going a little bit deeper, they also took in the ano-rectal malformation patients, surgical specimens. Simply what they operated on was sent to the pathology, and a myriad of problems was seen throughout the layers of the intestine. General disruption, erosion of the mucosa, of the muscularis mucosa, submucosa, inflammation, fibrosis, everything's there. And they were all absent in non-ARM tissue. They also looked at nerve cells, and there they saw that the nerve plexi were sparse and concentrated into small parts in the submucosa and the muscular layer, but they also looked at the ganglion cells and the interstitial cells of Cajal, and they stained that with NSE C100 and c-kit. And in the Western blot and immunohistochemistry analysis, there was a marked significant reduction in these cells compared to the normal population. Another group repeated, in a way, the histopathological findings in these patients. So also, again, throughout the layers of the intestine, a myriad, again, of problems were seen. And they tried to compare the proximal part of the segment that was resected with the distal part that was resected. And they looked at the inner circular muscle layer, showing more hypertrophy in the proximal segment compared to the distal segment, and no true difference in the regarding the thinning of the muscle layer itself. Going a little bit deeper, then it's the other way around. The distal segment has shown more hypertrophy, and the thinner muscles were mainly seen in the distal segment. Maybe more interesting, in my opinion, is that the myenteric plexus is almost absent in the distal segment. Even in the proximal segment, only 40% of the population had a myenteric plexus there. That brings us a little bit to the sensoric system. So when looking at muscle spindle cells, which are proprioceptive, found in the muscle tissue, and they were found to be fewer in the puborectal space, both in high and low imperfect anus patients versus normal. There were no muscle spindle cells in the external anal sphincter. When looking at the Pacinian corpuscle, which is an exterior receptor located in the internal sphincter and the external anal sphincter, and also in the presacral space, which is only present in the presacral space in these patients, and some were found in the puborectal sling, and the free-ending nerves, which usually reside in the anal canal, weren't there. What about the motor neurons? Well, here as well, there is a reduced number of motor neurons in babies with anorectal malformation compared to normal babies and post-mortem tissue in the puborectal muscle, but also in the external anal sphincter. Also looking at the interstitial cells of Cajal, also reduced number in 7 of 12 patients, a reduced expression of these cells. There's a very nice studies done in a very nice rat model in which the ano-rectal malformation can be induced, and these studies were done and were throughout the pregnancy of these rats. They were operated on, and a tracer was given, and through the tracer they could also show that there was a reduced amount of sensory neurons in these rats with an ano-rectal malformation, also a reduced amount of motor neurons, and of course also a reduced amount of interstitial cells of Kajal. Those were most pronounced in the high type ano-rectal malformation, but also in the low type compared to the controls. So ultimately, I think looking at the studies done by Ms. Hikspoors, there's definitely more insight into the development of the cloaca, although of course the embryological development of the ARM is yet to be unraveled. The developmental and anatomical anomalies in the ARM are manifold, with seen within the pelvic muscle complex in the sensomotoric system, most pronounced in the unfavorable prognosis or the high type, depending on the study and how they were subclassified. May be related with outcome, of course not presented now, would be a different discussion, but it would seem logical that there is a relation there. And of course for the surgeon, it also means to be really aware of the anatomical variance, and that it's not simply the absence of a canal, but everything that is normally there, such as muscles and nerves, is there. And of course, a Merry Christmas to all, because I've heard that this is the last presentation of the season.
Indeed, it is. Thank you. Thank you very much. I would like to open up the discussion section of this webinar. If you stop sharing, you could maybe, yeah, you can then see the Q&A section, but I can see there's one question already about the biopsies that were taken. I think it was during your talk, Ruben.
Let's see. During the surgery, that explains the edema. Yeah, well, part of it, of course. So yeah, naturally, there are some different explanations as well. The tissues biopsies were taken during surgery, and the controls were from post-mortem tissue samples. So naturally, they are a little bit different, but I think things like fibrosis, like general shedding and erosion and stuff, I think that is not something that I would have expected as well in the post-mortem tissue as well. But it's a good question.
So for the attendees who still have questions, we have the Q&A section. I will point that out for you to know. It's and in the toolbar below, you have Q&A with a little question mark. That's where you can type in your questions if you didn't know already. So I will wait a couple of more minutes so you can type in your question if you like. And you can also raise your hand so we can open up your mic for a verbal discussion. In the meantime, I would like to mention that the webinar has been recorded. So I will send the link to the recording tomorrow via email. I will post it on our YouTube channel, and you can find all of our webinars on that platform, and also on our website. So make sure you subscribe to our YouTube channel so you can always get a notification when we post something. And for the attendees who have attended live, you will get a survey after the session has finished. And if you fill that in within 2 weeks, you will get the EACME certificate. I can see that somebody, oh, maybe another question. Yeah, you can read it in the chat. So at the very end, there is a question: are there main transcription factors in the development of the cloacal membrane?
I don't know because we have looked indeed to the observation histologically and do see a difference in the cloacal membrane on the ventral side, which is mainly the rectal plate with very thick layered skin, whereas the cloacal membrane is very thin. But yeah, I can imagine indeed now with the insights that we have gotten from our study, so that part is not growing and needs indeed to rupture in order to have the anal exit, that future studies indeed should allow us to also get more insight in what kind of transcription factors are causing this difference in growth, allowing the change of the cloacal membrane into an open structure. Yes.
Thank you. I myself am more into the bladder exstrophy kind of field. Do you also have, maybe by any chance, models, 3D models on how the migration works in the embryological phase for bladder exstrophy? Because I think it's kind of the same in the sense that the migration is different than a normal situation. But maybe you haven't researched that part.
We have at this moment not yet put a focus completely on the bladder because we first had indeed for quite a long period our focus on cloacal subdivision and then its fate. So the metaplasia that we have seen and the difference in growth between the female and the male urogenital components. But yes, we have from all the pelvises of our models, we have the bladder now also in our 3D software. We have the reconstructions and we have 2 PhD students who are at this moment indeed also looking at how the bladder is developed, what is the influence of the allantois, yes or no, because we do see that the allantoic area, for example, also does not hold the muscle layers which are surrounding the bladder. So, and also the epithelium of the allantois is different from the bladder, or which will be the adult bladder. The trigonum, for example, is a very interesting area, but I've at this moment not enough information to have an idea about malformations. Yes, but we do have an idea how the bladder will be built within the embryonic period. Yes.
Oh, very interesting. Looking forward to seeing all those results. I can see there are.
There's one question also for Ms. Hikspoors. Do you see embryological evidence of the puborectal muscle? Alberto Peña doubts its presence altogether.
What we do see is the levator ani at its whole. So we do not see a difference in the 3 components, which is always shown in the atlases. Also, if I would go to the dissection hall and would dissect the human cadavers, then we do see in levator ani, but we mainly at the moment, this say like we have a puborectal area and we have a visceral area, so the 2 other parts of the levator are already taken together because there are doubts about can you see them separately, but I cannot. You could maybe, if we put volumes on several regions based on topographic location, you could maybe say that the distal end of the vertical levator, that's a little bit more thicker, and that's the area where you would expect then the puborectal area. But histologically, I cannot see different layers because we do not have also, for example, at this early phase, the fascia layer. So we do not have the isolated muscles from each other. So with respect to that, my answer is no. To me, it would be just like the levator ani muscle and not all those 3 individual components of it.
Good, thank you. I think there is one last question. Do you know why, for example, Down syndrome children have arm without fistula? No.
That would be for me, I think, Jill.
To me, not known. I only can think about that if fistula is formed, if there would be still some growth in that area, so that you would have a connection indeed of that dorsal area to the ventral side. But if you would have an acute stop, so maybe the disturbance that is causing a differential growth problem, that's in these Down syndromes also affected, so that you would have an immediate stop over there, so that the subdivision of the cloacas quite abrupt, without having then at these items fistula. But I don't know if there are still a lot of variations seen in children with Down syndrome, or that you only have one type of ano-rectal malformation. That I'm at this moment, but I will look into it, because I think it's quite interesting if this type of children would have would show less variation between the ano-rectal malformations, because that maybe could help already in order to see what kind of factors are involved in the differential growth.
Yeah. I think that was all on the questions that we've got. If you still have any, there is one last question at the very end, I see. I'm not quite sure. I think maybe both of you, or maybe Ruben can answer that one.
It's a kind of, well, the discussion for part, of course, is of course, part is what do we see when we dissect a patient, but the other part is our conceptual thinking of how the pelvic floor works and what is the function of the puborectal sling of the levator ani and so on. And indeed, it's true that there's a debate about the functionality of certain aspects of the pelvic floor muscles, and is it really there? And also in discussions with Jill, is the internal anal sphincter, how does it develop, and is it more part of the external anal sphincter, or does it come from the gut tube itself? So the philosophy, well, philosophizing about it is very interesting, and where we today think, okay, this is our truth, could be different in 50 years. Doesn't 100% answer the question, more or less that I'm agreeing with Mahmoud Marij, that one could put some questions to the general theories.
Yeah. Thank you very much. I think I have mentioned everything that I wanted to mention as well. So think of the survey if you have attended live. That will help us continuously improve our sessions, and we'll get you a certificate. And I would like to thank you, the presenters, for the very interesting presentations. Really enjoyed seeing the 3D models as well. It made it easier to comprehend all the changes. So thank you very much, and thank you to the attendees. And I would like to see you in the new year, I think. Yeah, tomorrow we have our last Ukrainian webinar, and then it's done for this year. So happy holidays, and we will see you next time. Goodbye.